Hello, welcome to Biot Anime. Today we are talking about agrobacterium-mediated cell transformation. Agrobacterium are gram-negative rods that belong to the bacterial family, Rhizobiaceae, which are often found near soil level at the junction of plant, stem and root. Agrobacterium is used for cell transformation, mainly due to the presence of tumor-inducing plasmid called TI plasmid, which is present in the Agrobacterium tumefaciens, and they induces tumor called crown galls, and the root-inducing plasmid called RI plasmid, which is present in the Agrobacterium rhizogens, and will cause hairy root disease. Crown gall disease caused by Agrobacterium tumefaciens is characterized by unlimited plant cell proliferation, which makes them grow autonomously on synthetic media in the absence of hormones. They also contains one or more of unusual amino acid derivatives known as apines that serve as a source of carbon and nitrogen for bacterium. Now let's look into the organization of TI plasmid. The length of TI plasmid is up to 200 kilobits and molecular weight is about 1.2 into 10 raised to 8. The major regions of TI plasmid are origin of replication, oncogenic region, region for conjugation, catabolism of apines and virulence genes. Oncogenic or ONC region contains rooty locus and results in tumor developing abnormal roots. And shooty locus results in tumors with abnormal shoot tissues. An opine synthesis or OS region is responsible for the synthesis of unusual amino acid or sugar derivatives, or collectively known as apines. Opine synthesis is controlled by enzyme, octopine synthase, and nopoline synthase, at loci called OCS, and NOS, on, octopine, and nopoline, plasmid, respectively. Finally, opine catabolism is controlled by the loci NOC, and OCC, for, nopoline, and, octopine, catabolism, respectively. These NOC and OCC genes do not form a part of tDNA. Next, let us look into the virulence gene, or VIR gene. 40 kilobytes VIR genes are involved in the virulence of Agrobacterium. These genes are present in 8 operon called VIR-A, 2, VIR-H, which are co-regulated and thus form a regulin. Most of the operon except VIR-A, VIR-F and VIR-G are polycistronic. The VIR operons are not transcribed during normal vegetative growth, except VIR-A and VIR-G genes. That is, the genes for virulence are silent till they are induced by certain plant factors. These plant factors are acetosyringone and beta-hydroxyacetosyringone, which are produced when the plant cell get wounded. The third component necessary for tDNA transfer consists of a number of genes encoded by the Agrobacterium tumefaciens chromosome, including CHV, A, CHV, B, and PSC, A, or EXO, C. The genes CHVA and CHVB code for exopolysaccharide production, which is important for the attachment of bacterium to the plant cell. The gene CHVE encodes a glucose or galactose transporter that is important for binding specific sugars that act as VIR gene coinducer. Now, let us move on to the very important topic of our DNA technology, tDNA transfer. tDNA transfer begins with the introduction of bacteria into the plant wound. Wounding of plants is necessary, because it'll lead to the production of acetosyringone and beta-hydroxyacetosyringone that act as an inducer of VIR gene transcription in the plasmid. The acetosyringone and beta-hydroxyacetosyringone is sensed by the VIR-A protein. VIR-A also interact with another protein encoded by CHVE gene present on bacterial chromosome and is important for the binding of sugar co-induces. The presence of these sugar and phenolic co-induces results in the autophosphorylation of VIR-A, which then transfer the phosphate group to the VIR-G, which leads to the activation of VIR-G. The activated VIR-G protein will then act as transcriptional activator of the other VIR genes. Early event in the tDNA transfer is the nicking of TI plasmid. 2 of the protein encoded by VIR-D, VIR-D1 and D2, provide an endonuclease that initiates the transfer process by nicking tDNA at specific sites. Or the nicking takes place between the 3rd and 4th base of the bottom strand of each 25 base pair. Repeat. Immediately adjacent to right border, outside tDNA, there's another short sequence called overdrive, which greatly stimulates the transfer process by functioning like an enhancer. VIR C1 and C2 will act at the overdrive sequence. VE2 will bind the single-stranded tDNA and is involved in transporting tDNA into plant cell nucleus. Most of the protein encoded by VE operon are located in the membrane, and these protein may form a conjugal pores, or pilus structure, in the membrane, through which the tDNA is delivered into the plant cell. VE11 gene has an ATP binding site, and the protein have ATPase activity. It may therefore involved in delivering energy required for tDNA transfer. VE operon consists of two genes, and it has a role in the detoxification of certain plant compounds that might otherwise adversely affect the growth of Agrobacterium. VE plays a role in tDNA delivery rather than symptom formation. Now let us look into how Agrobacterium is used as vectors. There are two types of vectors are found using Agrobacterium. 1. Co-integrate vector. 2. Binary vector. Co-integrate vector contains disarmed T region, origin of replication, virulence region, and bacterial selectable marker gene. The disarmed T region contains left and right tDNA border, terminator, plant selectable marker gene, 2 promoter region, and gene of interest. Binary vector consists of 2 plasmid, as the name suggests, plasmid that contain site for the insertion of the gene of interest, and a helper plasmid. The regions in the plasmid with gene of interest are tDNA with multiple cloning site, plant selectable marker, along with left and right border. It also contain origin of replication for Agrobacterium and E. coli. The helper Ti plasmid contain VE gene and an origin of replication. Thank you for watching the video. For more videos, please subscribe to this channel and press bell icon. See you again in the next video.